Alright, so this is the second one. I was just walking the dog after I made the first one, thinking about how that other one went. I know it was pretty quick, so if I could just draw like the chromosome stick diagrams here to maybe help this out a little bit more. So this is a pair of homologous chromosomes, and if these two genes were linked on the same chromosome, it's going to look like this, right? And if you were true breeding for wild type, right, that's the gray body, you're going to have gray, and then you're going to have red eyes and red eyes. So this would be the body color locus, and then this is the eye color locus. If they were on independently assorting chromosomes, right, then you'd have one pair of homologous chromosomes here, and you'd have gray on that homolog, on those homologs, and then the eye color one would be on this other different chromosome, red like that. So again, this is what it's going to look like if they were independently assorting. This is what it's going to look like if they are linked, right? So when this, well, let's go to the pardon me, to this cross then, this first cross that they're describing, right, is that true breeding fly with the true breeding black body and cinnabar eyes. So again, you know, that one, if they were linked, this fly over here, right, is going to have the black body, right, and then the cinnabar eyes there. And again, if they were independently assorting, right, then it's going to look like this, where you'd have the black body on that one, and then on the shorter chromosome here you'd have the cinnabar eye bit here again. So this is what they mean by independently assorting, and then this is what it would look like if it was linked. So when either way though, this is going to end up getting a chromosome that is this, right, in the gamete, right? And then if it was independently assorting, the gamete would look like this. Over here, there's one of the homologs in here, and this would be the G, and then this would be the R, like that again. So this is the result of, you know, independently assorting, and this is the result of those genes being linked. Okay, so then over here, the gamete, all the gametes would look like this if it was linked, and then if the gamete is if they were independently assorting, you'd have the gamete that looked like this. Okay. And again, so that when it comes to this F1 cross here, these flies in the F1 generation cross with true breeding male flies, black bodies, right? So if you go back and look at the problem again, like all of these F1 flies are going to be heterozygous at both loci, right? So they're going to be this, and then that gray is linked with the red, and then the black was linked with the cinnabar, right? So again, if these were independently assorting, this is what it would look like, right? You'd have heterozygous at this locus, and then on another different chromosome, you'd have the eye color gene, and it would be heterozygous there, right? So again, this is the linked situation, and this is the independent assortment situation over here on different chromosomes. So the gametes that this fly is going to make are going to look like this, right? And going to look like this. This would be. So then you produce the similar gametes over here, right? Where you'd have this, and then you'd have this long one that is the B, and this one that is the cinnabar, right? However, the difference here is that you are also as likely to see these gametes produced, right? the G, and then with the C this time, and this with the B, and this with the R, right? So due to independent assortment, I would expect to see an equal number of all of these different types of gametes here, right? But in the linkage situation, I would expect to see these, right, much more frequently. However, I am able to produce this in this kind of situation if I had recombination happening over here on this linked side. So in order to get a situation where I had the gray with the cinnabar, right? So again, here's the gray with the cinnabar over here. So in order to get that situation, I would also get this black with the red, right? In order to get these gametes right here, this is going to be the black with the red. I will have had to have recombination happening right here between those alleles. Right? So you essentially, would take these chromosomes, and anywhere along here you'd cut and then switch those. So the C ends up with the G here, and then this red ends up with the B. The red ends up with the black, and the cinnabar ends up with the gray. Right? So you will get this sometimes. Right? You'll get that sometime, but the majority of them are going to be this, and the majority of them are going to be this. Okay? So maybe that helps a little bit more, showing those chromosome stick diagrams of how you get those different types of gametes. Right? So at any rate, if you get most of them being again gray with the red, you have this gamete, and then the other gamete is going to be the black with the cinnabar. Right? And this cross, you're crossing that with a sperm, right? And so then the resultant combinations here, right, get you a reuniting of the homologous chromosomes. And so then these are going to be gray and black, and this is going to be red and cinnabar, and this looks like parent 1, and then so this is the result of cross 1. This is the result of the other fertilization possibility where I get this homologous pair reuniting, and they're going to be homozygous at both loci. And this looks like parent 2. Okay? And again, you're going to see these most of the time because you saw these gametes most of the time. Right? In order to get the other recombinations, right, you would have to take this sperm, that sperm up there, and get the gamete that is the result of the recombination, and this is the other gamete that is the result of that recombination. Sorry, this is red down here, right? Red, and still fertilizing with this same sperm here, and so then this and this would be 3, this would be 4, and so reunite in fertilization, and this is going to be heterozygous, and it's going to be homozygous there. So this is not like either parent, that's a recombinant offspring. So this is the result of 3, and the result of 4 is reuniting the homologous chromosomes here, and this guy's going to be homozygous at this locus, but then heterozygous at this locus, and again this offspring doesn't look like either parent either, so these are the recombinant offspring here, and again you got these because of the result of crossing over to get this only sometimes and this sometimes. Okay, hopefully that helps. Again, if you have any questions, please ask. Bye.